Our group is B7, including Marshall Stauffer, Francina Jizu, Ron Ding, and Paige Goodham. Our topic is the shaping of modern human immune systems by multi-regional admixture with archaic humans.
This article discusses the HLA system in humans. HLA is the human leukocyte antigen and is a part of the major histocompatibility complex, or MHC. There are a large group of genes related to the immune system function in humans, and these genes reside on chromosome 6. HLA genes in MHC class 1, which are HLA A, B, and C, produce proteins that are present on the surface of cells. These proteins bind to peptides and display them to the immune system. If they are recognized as an invader, the cell will be instructed to self-destruct. Overall, the HLA complex helps the immune system distinguish the body's own proteins from proteins made by foreign invaders, such as viruses or bacteria. This study suggests that we owe credit to our ancestors' encounters with Neanderthals and Denisovans for our immune systems. Neanderthals and Denisovans are both extinct species that are branches of the human family tree. They may have inherited several HLA alleles from the Denisovans and Neanderthals.
This figure displays traces of HLA C*1505 and HLA B*7301 in the human genome. This suggests introgression between modern humans and either Africans or archaic humans. The origin of HLA B*7301 and HLA C*1505 could have come from Africa or archaic humans. After humans split into two groups, the modern group and the archaic group, a group of modern humans migrated out of Africa. This group then split into two groups, with one group eventually migrating back to Africa, while the other group admixed with the archaic humans. Modern humans acquired HLA B*7301 and HLA C*1505 through introgression. The distribution shown in Figure 1 is consistent with the introgression of HLA B*73 in West Asia, site of admixture between modern and archaic humans. Also consistent with introgression is the linkage disequilibrium between HLA B*7301 and HLA C*1505. HLA C*1505 is predominantly found in West and Southeast Asia. This data suggests that HLA B*7301 and HLA C*1505 originated in modern humans from admixture with archaic humans in Asia.
So a diversity of the alleles of HLA A, B, and C genes is advantageous to the long-term survival, to human long-term survival. This is because HLA genes are the immune system genes that help our cells recognize and respond to threats, such as viruses and bacteria. That makes the HLA gene one that is very diverse, having over thousands of alleles. Studies conducted on the HLA gene show that they are critical to long-term human survival because T cells must maintain a variety of these genes in order to prolong life by fighting against bacteria and viruses. So there were two alleles of the HLA gene being observed in this case study. There's HLA B*7301 and HLA C*1505. Further studies show that the nature of these alleles is closely related to that of subsets of chimpanzees and gorilla alleles. The lineages of these alleles are divergent and distinct from the lineage to which other human HLA alleles belong. So this suggests that these alleles must have been recently acquired and integrated into the modern human from interbreeding with an outside source. So the question here is, where exactly did these alleles, where were they acquired from? So when analyzing the data, the B*73 allele distribution is more prevalent in West Asia, suggesting that some sort of admixture occurred between modern humans and some other form of humans from that region. Similarly, C*15, another allele, is most frequently found in Southeast Asia. Still, the question of where the alleles came from hasn't been answered, but the high frequencies of the alleles in certain parts of Asia suggest that that they must have been acquired by humans in that region of the world. In Asia, linkage disequilibrium between these two alleles is only about 90%. Worldwide, about 98% of people carrying the B*73 allele also carry the C*15 allele. However, in Africa, the linkage disequilibrium reaches 100%. So this basically means that there are some people carrying one allele and not the other in parts of Asia. However, in Africa, Everyone found to carry the B star 73 allele of the HLA gene also carries the C star 15 allele, and 100% of the time. So the fact that that linkage disequilibrium of these genes is 100% in Africa shows that genes could not have originated from Africa as previously hypothesized. Therefore, it must have been acquired from archaic humans. So these results suggest that introgression occurred in East Asia and then over time spread outward, eventually reaching Africa. Also, the fact that the B star 73 gene the fact that it is absent in cohesion, speaking, and pygmy populations further strengthens this point because these populations migrated out of Africa long before modern humans migrated into and out of Africa. So the fact that these populations do not carry these two alleles shows that the allele could not have originated from Africa. If not, they would have those alleles. Further experimentation also points to the idea that the presence of the B star 73 allele in modern humans could have arisen from introgression between modern humans and Denisovans. Similarly, the presence of the C star 15 allele could possibly have arisen from introgression with Neanderthals. So basically, on migrating out of Africa, modern humans encountered archaic humans who lived in Eurasia for many years, therefore having immune systems better adapted to the local pathogens in that area. Such adaptations almost certainly involved changes in the HLA gene, and introgression between modern humans and archaic humans caused for modern humans to acquire the B star 73 and the C star 15 alleles of the gene. So basically, introgression between modern and archaic humans, along with natural selection, helped shape modern immunity because the acquisition of these alleles added to the diversity of the HLA gene, and these genes all fight against certain viruses and bacteria, therefore strengthening our immunity today.
In closing, according to recent data, it can be concluded that the possession of certain specific alleles that govern the immune system suggests a possible link between prehistoric humans and our modern-day selves, possibly due to interspecies breeding.